Good morning, evolutionary biologists. Thank you so much for being here for our SSE session. All right, my job here is to make a few announcements about some of the awards and activities that SSE is involved in before I introduce our president for her talk. So I want to go ahead and start with my first slide. There we go, thank you very much. First I want to acknowledge, hopefully you guys, it's on, can you hear me? Yes. First, I'd like to acknowledge our IDEA Award winner, Dr. Corey Welch. Hopefully you all were able to see his talk on Friday afternoon. The IDEA Award is given to a person of any career stage who has strengthened the ecology and evolutionary biology community by promoting the things we value the most, inclusiveness and diversity in our fields. I thought Corey did an amazing job of telling us a very personal story, but at the same time, also the impact of the kind of activities that he has been involved in his career. So thank you, Corey, for putting the real effort into those areas that we value so much. Thanks. Okay, the next award SSE gives is the Huxley Award. This is run by our SSE Education and Outreach Committee. And this award recognizes and promotes the development of high-quality evolution educational resources, which we all know that we need very much, right? Our award this year goes to Tim Hartelt, who's a PhD student at University of Kassel in Germany. I don't believe Tim is here, but if so, please stand and let's give him a round of applause from across the world. Tim's award-winning materials were entitled Metaconceptual Learning Activities for Promoting Metacognitive Awareness and Self-Regulation of Intuitive Thinking in Evolutionary Education. All right. My next award are the President's Award. And if you notice here, President's is actually with the apostrophe after the S. And that is because it is the award of 3 presidents. The president-elect, Carol Boggs, the current president, and Charmantier, and myself, past president, Kelly Zamudio. And the President's Award, I have 2 awardees, an award winner and an honorable mention. And it is given to, chosen by the 3 presidents, and it's given to the best publication published in the journal Evolution during the preceding year that was contributed by a student and was part of their dissertation. So because I read all 30 of these papers, and all 3 of us read all 30 of these papers, we have a special connection with this particular award. And so if the 2 winners are here, I know that at least one of them is, I would like to call them up to the stage for a handshake, and I'll announce their names as they make their way up. The winner of the President's Award is Samarth Mathur for his paper. Samarth, come on up. His paper was entitled An Evolutionary Perspective on Genetic Load and Small Isolated Populations as Informed by Whole Genome Sequencing and Forward Time Simulation. Congratulations.
Thank you.
Hang tight there. Let's see if Robin is here. And is Robin Costello in the audience? You see? Perfect. Great. Robin won the honorable mention for the President's Award for her paper, Multi-Level Selection on Social Network Traits, Differs Between Sexes and Experimental Populations of Forked Fungus Beetle. Congratulations. Great. Thanks, you guys. Robin's paper. Great. Okay. The next award I'd like to talk about very briefly, because we are still going to have the opportunity to hear from these particular award winners. The next award the SSE gives is called the Dobzhansky Prize. It's awarded annually by our society to recognize the accomplishments of an early career researcher with future promise of an outstanding career in evolutionary biology. Our winner this year is Chloe Schmidt. She will be giving her talk today during the SSE Dobzhansky Prize session, which is also combined with the ASN Career Awards, and that is at Sunday, today at 2:30, so please attend her talk. Let's have a round of applause for Chloe. We here at SSE seem to be either very indecisive or also super inclusive because we have honorable mentions. We just love our honorable mentions. Dobzhansky Prize also has an honorable mention, and so I want to make sure and acknowledge Kim Huang, who is the winner of the honorable mention for the Dobzhansky Prize this year. So, Kim. Yeah. So Kim is also speaking today, not in the Dobzhansky Prize, but in a contributed talk. So please find her for that talk. Great. Okay, next up, we have the W.D. Hamilton Award. So this award is given by SSE for outstanding, the best outstanding graduate student presentations. The W.D. Hamilton talks were given during the virtual component of our meeting a few weeks ago and are judged by a committee of SSE members. And our overall winner today was Mackenzie Urquhart-Kronish for her talk in the Hamilton session, and we had 2 honorable mention follow-ups, Yuki Haba and Michelle McCauley. If all 3 of them, I don't know if any of them are here, but if you could please stand so we could acknowledge you. Congratulations. Okay, for this next award, I am going to turn it over to Emily Snell-Rude, who is going to make the introduction of our Lifetime Achievement Award.
All right, good morning. It is my honor to introduce my colleague Ruth Shaw as the recipient of the 2024 SSE Lifetime Achievement Award. Over the last 40 years, Ruth has made huge contributions to evolutionary biology in her research, her support of students, and her service to the society. Ruth has developed and tested foundational methods for measuring fitness in natural populations. For instance, Ruth's Aster models go beyond just measuring single traits at a single point in time to look at fitness over the lifetime of an individual. She has moved the theory into the field by measuring fitness of individuals in natural populations. For instance, prairie flowers. Often her individual measures of fitness span over a decade. These long-term field experiments reveal trade-offs and correlations that are often obscured in the lab. She's also made fundamental contributions to understanding the origin of novel mutations, for instance, finding that de novo mutations often are less deleterious than assumed. Her work has implications for understanding responses to environmental change, for instance, thinking about how genetic variation and genetic correlations may limit responses to climate change. She's not only built bodies of empirical work and conceptual understanding, but also entire toolkits and for instance, collections of seeds for research and conservation. In the realm of graduate education, she has taught a course at the University of Minnesota in quantitative genetics for over 30 years, where she seeks to teach the why behind the equations. She has advised over 20 PhD students and 14 postdocs and served as the director of graduate studies in our program. In SSE, she has served on the editorial board. She's been the editor-in-chief of Evolution. She's been on the council. She's been on the finance committee. She's been vice president, and she's been president. So please join me in thanking Ruth for her decades of contributions to the field of evolutionary biology.
Thank you all. I am truly overwhelmed and deeply honored by this award. In seeing you all and appreciating this as I do, I am reminded of how overwhelmed I felt at my very first evolution meeting so many years ago, probably 40 or 43 maybe, my second or third year as a graduate student, when a few of my peer grad students were brave enough to approach the evolutionary biologists whose published work they so respected, but I was not. But this community has been such an inspiration and support for me for all these years, and I thank you.
All right, thank you. All right, so now it is time for the talk by our president, and it is my pleasure to introduce Anne Charmantier. She is the president of SSE, and we'll be giving her address. Anne is an evolutionary ecologist who's interested in organisms and how they adapt to their environments. She's especially interested in cases where this involves some sort of speed or some sort of rapid type of environmental change. Her focus has been in the past on birds, but not exclusively, and even within birds, the overall range of her research questions is incredibly broad. So she's interested in organismal phenotypes and their responses, everything from life history to morphology, behavioral traits, and I think the trademark of all of this is that she's interested in all these phenotypes in natural populations. She has been recently focusing a lot on the evolution of these phenotypes like phenology, diseases in response to stressors, global stressors such as climate change, and also stressors such as urbanization. She is very active in service in our community. Not only is she president of SSE this year, and she will be past president next year, she's also the chair of the Equal Opportunity Committee for ESEB, and is in both those roles here at this meeting, at this joint meeting. And I was talking to her yesterday, and I learned about her active mentorship in a program in Montpellier for women in science, where it sounds amazing, but every woman PhD student at Montpellier is paired with a mentor. So it's a mentorship program at an incredible scale. Given her research in global change stressors and organismal responses, you can imagine that she does a lot of outreach, especially to news and media outlets. I also note that she has shared in a publication of her personal experiences and what they mean in terms of her motivations for advocating for people with disabilities in STEM. And in working with Anne for the last year, what I have noticed is that she brings her whole self to everything that she does, and she serves really as a role model, as a leader in terms of our community and how she leads with intentionality and also with empathy. So it's been really wonderful for our community to have her as a president, and I invite you, Anne, please, to come up and share your experiences with us.
Thank you so much, Kelly. If I was stressed before the introduction, not sure what state I'm in now. Quite emotional. Okay. So thank you, Kelly, yes, for introducing me, and thank you all for being here. I know that Montreal is a super great city to party, so I'm grateful that there are so many of you in the room this morning. It's a truly immense honor to be here as SSE president, and I have to say I thoroughly enjoyed the last year and a half getting to know the SSE from the inside, working with amazing people in the executive and council committee and meetings, and getting to understand and participate in all the efforts that are being made to make evolutionary biology a diverse, inclusive, dynamic, and vibrant community, hand in hand with our sister societies, of course. So I can't invite you to come to the SSE Members Forum because it happened yesterday, so thank you for those that attended. But I can tell you, at least, that this year, in all the efforts that are being spring this year, one of them I want to talk about this morning is the initiative to launch a strategic planning committee. And so I welcome everyone who wants to come and talk to me about this. If you have ideas about the priorities that should be for the future of our society, do grab me at coffee break or in the corridor. I'd be very happy to talk about this. So I've chosen this morning to give a scientific talk about a field that I'm very excited about, and that's urban evolutionary biology. But first I thought, coming from across the pond, I think that many of you don't know me, so I thought that I start with a little bit of an introduction of who I am and where my inspiration comes from. So these are snapshots of my childhood. I was a French urban kid spending most of her weekends and holidays in the wilds, mainly hiking. And I think a very important factor during my growth that was really determinant for my choice of career, and that is thanks to my parents and the education they gave me, is that I felt I was a part of nature. And also nature was springing a lot of questions, it was nurturing a lot of questions in my head. And these two things are things that I try to reproduce when I do outreach and I meet with students and children. And here I want to pay tribute to Canada. So Canada is the country hosting this conference, but it's also the country that hosted my family for several long-term stays when I was a child. And I think experiencing the wildness and the wildlife, the amazing wildlife of this country was really a trigger in my life. So after one of these stays, I was 11 years old, we had spent a year in New Brunswick. I came back to France convinced that my life goal was to study whales, or perhaps polar bears. But there you go, you know, how a motivation for science emerged in this country. And then finally, as a tribute to Canada, I also want to mention that this is the country where I have the strongest collaborations outside of my home country of France, in particular with my long-standing colleague and friend, Denis Réal. I'm not sure he's around, but he works at UQAM. Hi, Denis. So yeah, I'm very grateful to be giving this talk in Montreal. It really resonates for me to be here to give this talk. So, nurtured by nature during my childhood, I later hatched into the academic world, and my path to fledgling as an independent researcher was very much influenced by these three mentors I want to acknowledge here, because without them, I would not be here, for sure. So first, Jacques Blondel, who taught me everything I know about birds. Second, Leske Krook, who taught me everything I know about quantitative genetics. And then Ben Sheldon, who taught me how to survive in the academic world and also had great insight on birds and quantitative genetics. Okay, so first scoop of my talk, I didn't study whales, and also I didn't study polar bears, but I did get to start as an early career evolutionary ecologist in this type of pristine landscapes. So let me give you a little bit of a taste of this place. Okay, the sound doesn't work. Ah, shoot. Never mind. You should hear the birds of the... So we are here in the forest of Piriot. It's in a man and biosphere reserve called the Fango Valley. It's in the northeast of the Mediterranean island of Corsica. And this is where I spent my springs doing fieldwork during my PhD in the early 2000s. And I spent many, many springs later on in this forest chasing birds until events in my life made it too difficult to have fieldwork in such remote places. So then I shifted a little bit the landscapes for some of my research, and this is the story I'll tell later on. But it's in these pristine areas of Corsica that my collaborators and I have explored many questions about evolutionary and plastic responses in specifically two model species, the blue tits and the gray tits. And especially we've been studying in the last 2 decades how these birds adapt to climate change. So we do so by using long-term data sets of birds that breed in nest boxes, and we use these data sets to optimize the way that we measure plasticity, evolution, natural selection, and heritability. And we model the processes that underlie these parameters, such as gene flow between different patches of woolens, genetic drift, or genetic and epigenetic mutations. And we are particularly interested in the complexity of the environment, the fact that the landscape can be fragmented, can be heterogeneous in time and space, and also the environmental stressors such as climate change. Okay, so complete change of scenery and complete change of atmosphere. This is a view of my hometown of Montpellier. And if I go back to this general framework, what we have gradually realized in the last 2 decades is that when one is interested in rapid evolution and plastic responses, cities are a really exciting place to study all these processes. And this is because cities have a concentration of factors that make them interesting and possibly affect the processes. So I don't think you can see my laser pointer, here it is. So cities are highly fragmented, they're highly heterogeneous, and they have all these what I call environmental stressors. So you know that cities are warmer, that's the urban heat island effect. There's air pollution, water pollution, disturbance by humans and pets and cars, light pollution and sound pollution, to name a few. So what I'm going to do this morning is that I'm going to give you a little bit of an insight into this field of urban evolutionary ecology with 3 different sections. In the first section, I want to explore what we know about natural selection, specifically in cities, and how different is natural selection in cities compared to more natural areas. In the second part of my talk, I'll tell you about our long-term studies of gradients along urban gradients. And then in the third, shorter part of my talk, I'll tell you about how we increasingly integrate the human social factor into our eco-evo studies. So what do we know about natural selection in cities? Well, in this framework that I've been outlining, studying eco-evolutionary processes in urban contexts, I think there is one consensus emerging from the literature, at least in statements that people give in their review papers, as I will show later, and that is that we expect the shape and the force of natural selection to be very different in cities from natural habitats. Very often we actually expect natural selection to be stronger in cities. Now, if we take a step back on what we know of urban ecosystems, I want to talk about the field of urban ecology. It's a much older field than urban evolutionary ecology. It emerged in the 1950s, and here I'm giving you an overview of some of the books that have been published, from the 1977 book by Barry and Casarda to one of the latest books published by Pedro Barbosa in 2020, Urban Ecology. And this extensive literature on urban ecology describes how cities influence species diversity, community composition, ecosystem functioning, interactions between different species, and especially between people and nature, impacts of cities on biodiversity and also on human health and well-being. And also most of these books have chapters developing urban conservation projects. Now, in terms of evolutionary processes that I mentioned previously, they remain very elusive in these books. Natural selection is cited very often in the way that, for instance, Kevin Gaston cites it in his 2010 book, and I quote here, "The urban environment represents a mosaic of novel ecosystems where natural selection operates differently compared to non-urban settings." Okay, so they seem to all agree that natural selection should be different. Now, in the last decade, however, urban evolutionary biology has truly emerged with a rising interest in this processes of natural selection in cities. Here's an overview of some of the perspective, review, opinion papers that have been published since 2015. And this field is emerging thanks to the solid knowledge based from urban ecology. Now, one paper in particular was very interesting for me in terms of discourse around natural selection, and this is a paper published by Vincent Fugère and Andrew Henry in 2018, because they explain, so the paper is entitled "Human Influences on the Strength of Phenotypic Selection." They were interested in this paper in how natural selection is changing in areas that are disturbed by humans, so anthropogenic disturbances. So it's a larger scale than cities, but they clearly predict that anthropogenic disturbance should increase natural selections for 2 theoretical reasons. The first one is illustrated here. It's that we expect that these disturbances will create a shift in the adaptive landscape. So you see here the trait frequency of fitness, and these disturbances will create a shift here that usually leads to maladaptation, meaning that natural selection for the optimum phenotype may become stronger. And the second theoretical reason is that we expect in these areas a mean absolute fitness to decrease, and this will come hand in hand. So this decrease in mean fitness will usually come with higher variance in relative fitness, and that means increased opportunity for selection, or here, selection strength on the y-axis. So what Fugère and Henry then go about doing is that they decided to review the literature and test whether these predictions were true or not. They collected more than 1,300 phenotypic selection coefficients from 37 species, where selection was measured in population exposed to anthropogenic disturbances, so again, larger than the urban scale. And what they found is that indeed, natural selection is stronger in some population, but this is true only for some harvested populations and specifically fisheries, where natural selection can be very strong, especially for growth traits. In most of the other cases, what they found was natural selection is most often weaker, and this is because of decreased opportunity for selection. So in terms of weaker selection, I wanted to highlight one paper where this was found, a study on the northern cardinal by Rodewald and Arcese. We're coming back here to the scale of the urban landscape that I'm more interested in. And what Rodewald and Arcese write in 2017 is that urban breeders might also be expected to experience relaxed selection pressures if urban environments are buffered from deleterious effects of environmental variation experienced outside cities. And these deleterious effects may be, so I'm giving 3 examples here. It might be that in cities you have a higher abundance of food resources of anthropogenic origin. It might be that you can also have reduced or even an absence of predator. And I've cited the urban heat island effect previously, which may create warmer conditions that are favorable for some organisms during their growth. So this can actually lead to relaxed selection. So you can see that in the end, in the literature, there's no consensus as to whether we really expect stronger or relaxed selection. So we decided in my group about a year ago, I think it was in May, June 2023, that we were going to review the literature, specifically the urban literature, to understand whether natural selection is really different in cities, and if so, how? How does the urbanization shift the shape and the force of natural selection? And so we did a literature review. We thought that we would find many examples showcasing weaker, stronger, or reverse selection. Also, of course, we reviewed papers where they didn't find any difference. And so we were specifically looking for quantitative measures because our aim was to conduct a meta-analysis. So we gathered all the selection gradients and coefficients, selection gradients and differentials that we could find at the phenotypic level and at the genomic level. I'm not going to talk about the genomic stuff because there were very, very few estimates, even less so than the phenotypic ones. And so we wanted to find studies where these gradients and differentials had been estimated in both urban and non-urban populations so that we could run this meta-analysis. But we found only 8 studies that had this kind of selection coefficients. So not enough to do a meta-analysis, but it was very, nevertheless, very interesting to read these studies and understand their results. So I've classified here these 8 studies, and I've added also studies here. The dotted lines are studies that didn't provide selection gradients or selection coefficients, but did have some really interesting results on how natural selection was, even if it wasn't quantified in a way that we could put in a meta-analysis. So I don't want you to look at too much in the details. This is just an overview. You can perhaps read the paper whenever it's accepted. But what I wanted to do is detail 2 examples to showcase how these studies are developed. Okay, so the first example is on the yellow jessamine, and this is work by Rebecca Irwin and colleague, especially in the city of Raleigh and other cities in Durham County, so it's in North Carolina. And what Rebecca Irwin and colleague did is that they measured in the field several traits on this Gelsemium species in 8 paired forested and urban and non-urban sites. They measured flower size display, but also several other traits relevant for flower size, flower shape, and also an alkaloid gelsamine resistant trait. And this graph presents the results on natural selection. So you have here the linear selection gradient for the different types of traits. The PC1 and PC2 are these different measures of flower size and shape. And what you can see is that overall selection is very weak, except for this one trait here, flower display, where you have strong positive selection, and this selection is stronger in cities. So urban flowers are strongly selected to be larger. But I do want to say that one of the conclusions here was that urban landscapes do not result in sweeping differences across phenotypic selection, but rather one strong result on one trait. The second example I want to showcase is one of my favorite. It doesn't provide any selection gradient or coefficient, but I think it's a really interesting study conducted on eastern gray squirrels in the city of Syracuse, in the state of New York, and other cities in New England. And this study by Bradley Cosentino and colleagues was based on the observation that in these cities in New England, you may notice that the eastern gray squirrel is sometimes not gray, but it's black. And so if you go further away from the city, this melanic black morph is completely absent. There are no black morphs in the forest. But the closer you get to the city center, the higher the proportion of this melanic form becomes, and it actually constitutes 50% of the population in the very center of these cities. So what Cosentino and colleague did is that they did a translocation experiment. So they collected gray and black morphs of the eastern gray squirrel, and they translocated them in urban and rural habitats, and then they followed the survival of these squirrels. It's quite obvious from this graph that when you are in a rural habitat, there is a strong survival selection against the melanic morph that are dying much quicker than the gray morphs. But in the urban areas, this survival selection disappears. So you no longer have an advantage of being camouflaged as a gray squirrel. So this relaxed selection against the non-cryptid melanic form is most probably because of the lower predation rate and absence of hunting in cities. Actually, I want to mention, I've written it here, that there are two talks about this project here in Montreal. I think one of them was yesterday, but there is one talk today, so I will check it out, and I'll see you there if you come. So overall, what we found with this literature review is that we really lack estimates to give a quantitative answer on how natural selection in city shifts in terms of shape and in terms of force. But we do find really interesting results highlighting that the answer probably depends on the traits you're studying and the species you're studying. And also, I didn't talk too much about this, but we're trying to really uncover what are the drivers of the differences in natural selection. And this is going to be a challenge ahead. But I'm pretty sure that many more people have data to quantify natural selection in cities. So this is kind of my call to run and publish these analyses so that we can make quantitative estimates and meta-analysis in the future. Okay, so let me now introduce you to our work on urban and non-urban great tits. And I will do so by using this roadmap. So this figure was published in a book that was edited by Marta Silken, Jason Muchin-South, and myself in the worst possible year to publish a book, 2020. No conference in 2020, no conference in 2021, so this is why I'm allowing myself to advertise for it here. And in this book, we wrote a chapter with my postdoc Charles Perrier and PhD student Aude Kézergue, entitled Adaptation Genomics in Urban Environments. And this chapter was proposing this roadmap to how to go about studying adaptive evolution in urban environments, but really it can be adjustable to any kind of environment where you can find differences. And here my leitmotif example is the pepper moth and the melanic morph. So usually our studies start with observing different phenotypes. And as an evolutionary ecologist, the first two questions that spring to my mind when I see this kind of dimorphism is first whether it's adaptive, and second, or sometimes first, whether it's of genetic and plastic origin. And so I'll show how we've addressed these questions in the great tits. And then also, if you find a genetic or plastic, usually both type of answers, you can also dive into, let's say, the genomics era. And you can do so in three different ways. Either you have one specific phenotype that you know is associated with the candidate gene, so you might want to target this gene. Or you can do, and this is what we've done, use approaches that reduce the genome complexity, such as RAD sequencing. Or if you are lucky enough to be funded by a princess or a prince, which is not my case, you can run whole genome sequences on your individuals. Okay, so I'm going to adapt this roadmap to the developments of our results in the great tits. But first, I want to say that as all long-term field projects and as all my research has been always the case. This is very much a collaborative project. Nest boxes were erected in the city of Montpellier by Arnaud Grégoire and Marcel Lambrecht in 2018. I followed a year later, and then these are some of the people that were involved in this project in the past years, and most of the results I will present today are from the work of Charles Perrier as a postdoc and Aude Kezerg and Megan Thompson as a PhD student. Megan defended here in Montreal 2 days ago, so it's been an ever exciting week. Successfully, of course. Okay, so as I said earlier, this work is based on one essential, very sophisticated tool, and that's the nest box, our star tool here. And so here you have an overview of the forest site that we have outside of Montpellier, around 30 kilometers northeast of Montpellier, the forest of La Rouvière. Each little dot, if you can see them, is a nest boxes. This is a closer view of part of the forest. And this is also a closer view of one of our urban sites. You can't really see the dots and the numbers here, but what was important for us is that we put nest boxes in different areas, and in each area we had nest boxes in parks, but also along roads. So we have a full gradient of urbanization here. Yeah, these are the nest box numbers, and I've written between 200 and 250 in the city because there's a difference between working the forest and the city, is that in the city there's kind of an arms race between researchers putting up nest boxes and nest boxes disappearing. So you have to put up nest boxes every year. So that's why numbers fluctuates a bit. And this is an overview of the 8 sites that we have in Montpellier, which, as I said, have different urbanization within the site, but also across the site. And you see the gradients here, that there are some sites that are very natural. Here is the zoo of Montpellier in the north, and then some sites that are very, very urbanized. So an overview of how we collect data. It's a very basic monitoring of reproduction. So our fieldwork runs during the whole breeding season from March to July. We actually ringed the very last chicks 2 days ago. Well, I didn't because I was already in Montreal. So every parent and every nestling is captured and ringed with an individual ring, which is very important for my quantitative genetic approach. And then we measure morphological traits. So we measure size via tarsus length, we measure body weight and relative wing and tail length. This is a blue tit being weighed, and you can see it weighs 9.4 grams, so it tells you how light these birds are. We also observe life history traits, especially the first date that an egg is laid, so that's laying date, the number of eggs, number of hatchlings, and number of fledglings. And then we have several measures on behavior and cognition. And in particular, I'm highlighting here because I'll show a little video later on, the test for the exploration score. So the way it runs is that you capture the bird, and immediately after capture, it's put in a little cage here where it can rest for 2 minutes. And then it is released in this part of the cage here, where it can explore this novel environment. Meanwhile, these are lights that control for the light condition, and here, a little bit hidden, is a camera filming the bird during this 5-minute trial of exploring a novel environment. And you'll have a little bit of the inside view a little later on. Okay, so now the results. So let's start with what we found in terms of phenotypic differences between the urban and the forest birds. So first of all, the summary of this part of the result is that every trait that we measured, as soon as we had enough power, after 5 or 6 years, sometimes 10 years of measures, we found that city birds were different. So for a start, city birds are smaller, they are lighter, and they have shorter wing and tail lengths. In terms of life history, they breed around 4 days earlier. I'm giving here the first egg dates for 2024. As you can see, 12th of March in the city, 26th of March in the forest, quite a large difference. Also, birds have 2 eggs less on average in the city. They have a smaller number of hatchlings and smaller number of fledglings. And then in terms of behavior, they display a higher aggression, faster exploration score when they are placed in this novel environment, and they have faster breath rate under constraint. Now, I've shown these results as comparisons between forest and city, but we also ran these analyses as gradients, and we find the same results along the urban gradients within the city. And then importantly, I want to say that in the last few years, we have reproduced these results, so not only in Montpellier, but in other cities. You may be surprised, those of you that don't work on blue tits and gray tits, to learn that there is a database called Spybirds where you can share your data sets, and there are 70 different data sets on blue tits and gray tits. So there's a lot of us out there studying these species. This is probably why it's a model species in ecology and also evolution. And so Megan Thompson, during her PhD, was able to gather 13 data sets where they had measures on both urban and non-urban birds. And she showed that a lot of the results I presented here, especially the fact that birds are lighter and smaller, but we also studied life history, they are similar across all these, mostly similar across all these cities. And then also, I will not dwell into it, but we have ran quantitative genetic analysis using the long-term pedigrees, and we know that these traits are all heritable. So that means that we're in a situation where we have strong divergence in traits that we know are heritable. So I think it's a fair question to ask whether this is the results of adaptive processes and whether natural selection can explain these shifts. So testing natural selection was the job of Odke Zerg in the early stages of her PhD. And I'm going to show a little bit briefly, but I'm going to walk you through the results so that you can understand them, what Odd found in terms of natural selection. So as a reminder here, I've told you that birds are smaller in the city, and here I'm showing you selection gradients on the different morphological traits. So actually, many of these gradients were non-significant, so I'm going to focus on the two that were significant. When you have a positive selection gradient here, it means that selection is favoring taller individuals. The round symbols are male, triangles are female, and the green are forests, and the purplish are city birds. So here we find that selection is favoring taller males in the forest, and also selection, and I should say these results are on reproductive selection. So selection is also favoring lighter males. So in the forest, lighter and taller males have more offspring. It's not the case in the city. So it doesn't seem to be aligned with the morphological differences that we found. Now, in terms of life history, as a reminder, again, birds lay earlier and smaller broods, as I told you before. And yet, what we find is that even though they lay earlier, there's no selection to lay earlier in the city, but there is strong selection to lay earlier in the forest. And even though they have smaller broods in the city, there's actually strong selection to lay larger broods in the city. So you're starting to understand where I'm getting at here, even though at the start we were a little bit puzzled. And then just giving a summary, because the graph would be too complicated, but we found, so we did selection analysis via reproduction, but also survival. And what we found for behaviors is that all the behaviors that are displayed by urban birds, so the fact that there are faster explorers, more aggressive, they're all associated with lower survival. So overall, all these results were a little bit surprising for us, but they seem to show that the observed phenotypic divergence that I described before are not due to an adaptive response, so they might actually be signs of maladaptation. Okay, so moving on to the second major question that, I had finding this difference in morphotypes and ecotypes, is whether these differences are of genetic or plastic origin. And here I'm only going to give a teaser summary. This is because Megan Thompson is going to give a talk Tuesday entitled "The City and Forest Birds Flock Together in a Common Garden Experiment." So she's going to give you all the details about this crazy experiment, but I can't resist and tell you a little bit about it because, well, I think I was warned by people working on vertebrates that I should not try and attempt a common garden, but we did in 2022. I'm going to show the results here, some of them, and we did it again this spring. I'm not sure why we insist, but perhaps because I think the results were promising. So what we did is that we collected eggs along an urban gradient, 50 eggs in an urban gradient, and 40 eggs in the forest. These eggs were placed under foster parents in the zoo. It's an intermediate environment between forest and city, and the foster parents were in charge of incubating and raising the chicks in the very early stages when they don't thermoregulate. And then the chicks were brought to a nursery where we hand-fed them. And it's a huge work. It involved around 20 people. We had to feed them from 7 a.m. to 9 p.m. constantly, no pause. And then the last step is the phenotyping. So we measured all the traits that I showed previously, and we also started a new project on physiology. I'm not going to have time to discuss this today, but come and see me if you want to know about this. So I'm just going to show you 2 results, and the details, no stats here, all the details will be in Megan's talk. So here are results that I showed earlier, that we reran during the years of the Common Garden, and we found that indeed in the wild, urban birds here are lighter. So what happens with these Common Garden birds that were raised in the same condition? This is what happened. First of all, they're much lighter, and this is expected because they are in aviaries. It's not the wild, so they don't fly in the same way, and so that means that they don't develop their whole muscle mass. But we did find, and again, I'm not showing you the stats, but surprisingly, even with this small number, we found that there was a difference that was maintained in the sense that birds that originated from urban eggs were still lighter than birds originating from forest eggs, meaning that it's a possible genetic origin, or maybe very early maternal effects, but we also ran quantitative genetic models showing that maternal effects at this stage are very small. So a promising result for a genetic difference. And this is the inside view of the exploration cage that I showed you earlier, the open field cage. So this is a bird exploring the novel environment. And as a reminder, what we had found previously is that urban birds have faster exploration, meaning they have a higher number of hops and flights when they are placed in this completely new environment. What we found with the common garden birds is that this difference completely disappears. So most probably the differences that we see in these behavior are of plastic origin in response to the urban conditions. Okay, I'm going to end these results on great hits with the genomic approach that we took, and especially one study that we did in collaboration with Marta Sulkin in Warsaw and Juan Carlos Senar in Barcelona. And so we gathered blood samples from these 3 cities of Montpellier, Barcelona, and Warsaw. We had 10 urban and 10 forest birds from each area, and we ran 2 types of sequencing. So we had rat sequencing, which gave us 80,000 SNPs across the whole genome, and then we also studied RBS to have methylation sites with more than 1,700 sites per individuals of methylation. Okay, so I'm going to show you results as the redundancy analysis. For those of you that don't know, it's very similar to a PCA, where each dot here, each symbol is an individual, so a genome, and the closer 2 dots are, the more similar the individuals will be in terms of their genomes, here based on the 74-something rat sequencing across the whole genome. And I have the same kind of symbols as before. So you have the triangles are females and the circles are male, and the urban birds are full symbols, the open symbols are forest birds. Okay, so what we can see with this graph is that the first and second axis of variation is the geographic origin. So birds are clustered very evidently by their colors. You have here the Barcelona birds, here the Montpellier birds, and here the Warsaw birds. And it's kind of something we expected. We know that there's genetic structure at this kind of scale. But the third axis of variation was an urban-forest contrast. And you kind of see it here because, for instance, for the Barcelona birds, you have all the forest birds clustered on the right here and all the urban birds on the left. And this contrast, this genomic contrast between urban and forest bird explained 2.2% of the variance in individual genomes. So do we have an even stronger marker of urban on the epigenetic variation, where here are the similar results, and here the first axis of variation was sex. It's quite a classic result. So you have all the males here on the right-hand side of the with the first axis and all the females on the left-hand side. So yeah, we know that methylation patterns are very different between males and females, something that is found in humans and I don't know about plants actually, but at least in animals it's quite classic. And then you also have this clustering by colors, which is representing the second axis of variation here that you can't really see, but you do see the cluster of colors, so the different origins of these populations. And then the third axis of variation here is the urban-forest contrast. So we did find a significant effect of the urban or forest origin on this epigenetic variation, but it wasn't larger than the one that we found at the genomic level. So 1.9% of variation in the methylation patterns was explained by this urban effect, so very similar to the genomic one. And another way to see these epigenetic effects, this is a graph that I like to show. It's called a circus plot. And for those of you that are not familiar with it, it represents the graded genome. So you have here the macrochromosomes of the graded chromosome 1, 2, 3, and then the chromosomes become smaller and smaller until they are so small that we no longer know exactly where these sites are. And each red dot here represents a site that is hypermethylated for urban birds. So more methylation in the cities. Each blue dot represents a site that is hypomethylated for urban birds, so less methylation in cities. And from the inner to the outer circles, you have birds from Barcelona, Montpellier, and Warsaw. Two take-home messages I find in this figure. The first one is that there is no evidence for parallelism in urban-linked methylation, and this is quite clear. If you look at here, one site that is hypermethylated for urban Warsaw birds, you won't see this site replicated in Montpellier or Barcelona. This is the case for most of the differentially methylated regions that we've identified. And the second take-home message is that we were kind of expecting cities to be associated with higher methylation, and this is not what we find. There are as many red dots as there are blue dots on the graph. So overall, kind of a conclusion of this on this second part on gradients in urban areas. What we have found over 12 years of studies is that there is very strong phenotypic differentiation between forest and city birds and across the urban gradient. This differentiation does not seem to be explained by natural selection, at least not how we can measure natural selection right now. In terms of reproduction and survival. Maybe it was linked to past selection, which is more difficult to analyze. In fact, but I'm jumping here to the genomic part. If we look at the genomic traces of selection, well, we do see an effect of urbanization on both the genetic and the epigenetic diversity, and it shows that adaptation is probably a polygenic and largely non-parallel mechanism leading to these different genomic differences. And then finally, the common garden experiment showed that some, but probably not all of the differences that we see in the birds may be due to evolution. Okay, so now for the final, much shorter section of my talk, I would like to address this growing ambition to integrate social factor into our eco-evolutionary studies. So this is a figure that was published in a Perspective article, published by a collective of mainly North American researchers in 2021. And it depicts how social factors can influence eco-evolutionary dynamics in cities. For instance, you have here the fact that social processes such as political or economic drivers and decisions can impact evolution by altering, for instance, the connectivity among habitats or through selection of preferred phenotypes that may differ across communities within a city. And I think this is a very important message. Future studies on how evolution impacts biodiversity in the cities should really take into account these social factors for many different reasons. One aspect that I've focused on, and this is because, as Kelly mentioned, I've been doing quite a lot of outreach in schools, but this outreach was basically going to schools and talking about women in science, talking about my research, talking about climate change and urbanization, but it wasn't really built in an eco-evo project, right? So what I decided to do 2 years ago is that I submitted an application for a new project called Acacia, who started a year and a half ago. for adapting to cities and climate change and interaction. In this project, we have a strong eco-evo approach where we study climate change and urbanization together, but we also have an educational science project. So this part of the project is led by researcher in the educational sciences, in particular Aurélie Swang and postdoc Alice Kosker. And the hypothesis that they are testing is that science education based on spy nest boxes that we've put up in schools and the idea of learning by doing will reconnect children with nature. So this project was implemented in the school year that just ended. It involves interaction between us evolutionary ecologists, the educational science researchers, and also the education authorities of Montpellier and Paris. And we had 10 classes involved in Paris, 10 classes in Montpellier, with children of 9 to 12 years old. And what we did is that in the fall and winter, we went and put up nest boxes in all these schools, and they are equipped with a solar panel here so that the kids can see what's happening in the nest box with a camera. All the views of the cameras are connected via an app, so if one of the nest boxes is not occupied, the kids can see what's happening in the other schools. And that meant that during the whole field season, when we were doing field work, the children were observing what was happening. And they were observing from nest construction to egg laying to the chick. Actually, there's a little video here from one of the school of a parent feeding the nestlings. And that went along with the development of a lot of different projects, and the projects depended on the teacher. It could be science project, mostly science project, but it could be math, it could be biology, many, many things. So my colleagues in the educational science haven't analyzed yet the results of this experiment that we will rerun next year also, but I do have some preliminary findings that I wanted to share today. First of all, the teacher witnessed increasing interest from the children during this experiment, and what I find even more fascinating, increased attention span. They said at the start it was very difficult to have the children being silent and observing, even like in forest and stuff, but then that really grew during the experiment. Also, it allowed the teachers to introduce knowledge and teaching on ecology, and especially urban ecology. And I don't know about here in North America, but in France, this is not part of the curriculum. So it was an opportunity to add some scientific knowledge. There was a great diversity of outputs. I'm going to show a few examples in the next slide. And that was because the classes differed a lot in their interest and what they wanted to do with that project. So that was great. And then more of a personal remark. I thought that interdisciplinary studies were about combining genomics and ecology, but try and combine evolutionary ecology with educational science. It was a true challenge. It was really interesting to see how the approaches, the semantics, everything is so different that it makes it quite challenging to work together, but it's also very, I can't say that, but rewarding when you succeed. So yeah, a truly fascinating experiment on the scientific side as well. So here are a few outputs. The poem is about the great tit, the mésange charbonnière, but it's all in French, so it won't tell you anything, but it's about what they observed during the season. There was also work about scaling. One teacher worked a lot in physics, and she did a whole project on using, like, for instance, here, an egg and comparing to everyday objects to get the children to work on the scaling issues. Okay, so I want to have a final conclusion of 45 seconds. Bear with me. So here's my final conclusion that is looping back to the title of my talk. From the wild to the streets, evolution never sleeps. Back in the day, scholars hit the green scene, forests serene where the silence was clean. Fast forward now, things ain't the same. Humans got involved and changed the whole game. From the woods to the hood, life's adapting, it's understood. Nature finds a way, even in the urban play. From the wild to the streets, evolution never sleeps. Urban life's a hustle, but nature's got a plan, adapting and thriving in the midst of man. Birds are getting smaller, and now their broods are light, more aggressive, exploratory, they're ready to fight. From the woods to the hood, life's adapting, it's understood. Nature finds a way, even in the urban play. From the wild to the streets, evolution never sleeps. Thank you all, and thanks to my colleagues. I think we have plenty of time for questions. There are 2 microphones, microphone 1, microphone 2, and I'm very happy to answer any question. Andrew Henry.
That was an awesome talk. Thank you so much. I really like your idea that selection that's occurring right now doesn't necessarily reflect the selection that got them to where they are. So is there any way to introduce tits into a city where they're not and study selection? Because that would answer the question, yeah?
Yeah. Well, initially I thought that the first answer to that conundrum of past selection, the ghost of past selection, was going to be genomics, but it doesn't solve it all. And one thing that I have said that for a decade. One thing that I really want to do is try and have a study in areas that are not yet urbanized, and follow what's going to happen with urbanization. The problem is just human resource, right? That you need to have an even bigger group if you want to try and do that. But certainly, I think it would be fascinating to see all the different stages of a colonization, because we don't really have a clue, at least in these species, who's arriving. It's highly probable that phenotypes that are selected early on are not the phenotypes that we see now. There's probably succession of events, and natural selection probably has a whole dynamics during colonization and establishment. But we are studying great tits in areas of the city that have been urbanized for a long time, and it's a millennium city, so it's not the best place to do that, but thank you. Yes.
Thank you very much. I really wish you hadn't finished your talk. It was fantastic to listen to you. So I wonder whether you have had time to get information on longevity of the urban and rural birds, because and to explore the possible correlations between the clutch size and longevity. So that it may be...
Okay, thank you for this question. Yes, I didn't present the results, in fact, but we did do a CMR, capture mark recapture analysis, to look at survival. There are expectations in the literature. I didn't mention that, but one of my great interests is around the pace of life syndrome. And there are expectations in the literature, and there's been actually reviews showing that organisms in cities might have a slower pace of life. This is not what we find in terms of behavior. They have a faster exploration and they're more aggressive. So we also tested it. So we looked at longevity in the sense that we looked at survival and longevity. And what we found is that actually survival is very similar in adults between the forest and the city birds, but the city juveniles, so the 1-year-olds, have lower survival in the city. And this is all in line with kind of the idea that is emerging that, at least in our population, it seems that city environments might be sinks compared to outside environments, like the source sinks dynamic. Now, for the second part of your question regarding I guess, correlations between survival and, did you say clutch size? We haven't looked at that yet. We started doing correlation covariances approach, but we are starting now the quantitative genetic analysis, so it takes a long time to gather a big enough pedigree. So we're now at 12 years of study on these urban gray tits, and we are starting now this kind of approach. So maybe, in a few years' time, maybe next year or in a couple of years' time, I can give you a more detailed answer on that part. Thank you.
Thank you very much.
Really inspiring talk and really very integrative and really cool stuff. I was wondering, perhaps for explaining these differences between the urban and the rural birds, did you consider stress and also perhaps immunology? Because, I mean, the city's habitat is full of noise, it's full of pollution and things like that, which might impact the birds.
Yeah, thank you for this question. So I've actually, I mentioned this new project, Acacia, that we started a year and a half ago. And in this project, there's a very heavy part on physiology. It's not my expertise. I've done very little physiology in the past, but I started a collaboration with Samuel Caro. And we are starting to take a lot of physiological measures. So immunology is not one of our major aim. We're more looking at heat tolerance and respirometry measures. But yeah, we could, I mean, I'm very happy to have also new collaborations. So it's also a matter of funding and time. So if people are interested, you know, this is something that we could develop more. It makes sense for sure.
Thanks.
Thank you.
Hello, excellent talk, and it was really great seeing the way the students responded to observing the birds. But my question was, with the differences between the different populations across cities, I'm also curious if there's anything known about differences in like colonization time of when these populations first urbanized.
Yes, thank you for that question. First, I want to acknowledge I cannot see you guys. There's too much light, so sorry if I don't recognize everyone. Yeah, so one idea that we had initially was that doing these meta and mega analysis across different cities would be very cool because it would allow us specifically to feed in the models things like how old the cities are, the population densities and things like that. In the end, one of the challenge of the urban environment as a landscape to study evolutionary processes is that there are many different features that are influencing plastic and evolutionary responses. So it's very difficult to pinpoint which are the ones that are important. And as I mentioned, we have many different data sets, but data sets with urban forest comparison, we had only 13. So it was, you know, good enough for a start. We were really happy about that, but not enough to have this kind of approach. But it would be very, yeah, a great challenge for the future, perhaps not in a vertebrate. I think maybe studying the white clover is a better idea if you want to do this kind of approach. Thank you.
Hi. Hey, yeah, thank you for the talk. I just had a question about the urban gradients, and you'd mentioned in the Common Garden experiment that the zoo was seen as kind of like an intermediate between the forest and the city. I was just wondering, in terms of the gradients, have yourself or other people looked at the likes of, say, like a village to a town to a city, or is the gradient more outskirts of a city to the center and that kind of thing?
Yep. Yep. Just to go back to the Common Garden, what we wanted was that the eggs would be incubated by non-biological parents. So it didn't matter too much, but we did choose this environment because it was intermediate, but that was not the main feature. The main feature was to decouple the biological parents from the development. But regarding your question on, sorry, I got lost in my answer. What was the question?
It was just about in terms of urban gradients in the literature in general.
Yes, thank you. Yeah. So in the other, so we have some weaknesses in our approach in Montpellier, that we have one big forest and an urban gradient that is mostly in the city. Some of it is on the outskirts, but we don't have suburban areas. But in the 13 populations that Megan used, for instance, there was a large variety. So some people do have a larger gradient that goes from suburban areas. These birds are not found a lot in agricultural areas, so that's not usually not present in the data sets, but this differs a lot across studies. Some studies, so initially a lot of the studies that were started in urban areas were in parks. So people weren't really studying biodiversity in the streets, they were focusing on parks. So some of the studies we've been using were really only in urban parks. So that, yeah, that differs a lot across the different monitorings.
Yeah. All right, thank you.
Thank you. Yes.
Hello. Wonderful talk. It was quite striking to see the differences of the, some of the phenotypes when they were common garden in the urban and the forest birds, and the phenotypes went away. If I understood the sort of common garden conditions, is it fair to say that they are closer to a less stressful environment in terms of noise pollution, for example? And so some of the phenotypes change. And I guess my question would be, what would happen if we were to expose birds from the forest to high levels of noise pollution or sort of those like classical urban levels of high stress? Would they develop high levels of aggression and high levels of exploration? Or is
Yeah.
that mostly genetic at that
Yeah,
point?
that's a good question. So in the common garden, we chose to have, we didn't have gradients of environmental features. You know, for instance, we're interested in heat tolerance, and it would have been nice to, in the common garden, have birds that grow at different temperatures or different sound conditions, different light conditions. But obviously our power, like the sample sizes, doesn't really allow this kind of approach. Another way to approach what you're saying would be to do translocation experiments rather than common garden. So like cross-fostering, for instance, the problem is that birds fly. And so it kind of makes it difficult then to track them. So, you know, you can put a forest bird in the city and see how it deals with the sound and the light, but actually you're just going to lose that bird. Unless you build aviaries in different conditions, but then the captivity makes it actually quite difficult for the birds. If they're not raised in captivity, they develop a lot of stressful attitudes in aviaries. So I don't know what the best solution is. Again, perhaps testing this on another model species than the great tits. You know, sometimes you just have to acknowledge that your model system is great, but it can't address all the questions you want.
Thank you.
Thank you.
Hi, thank you for your talk. I was wondering if there are differences between native and exotic species in terms of their capability to adapt to urban environments.
Yeah, certainly. So we know, I mean, this is more like a species, studies across species than like my research is more within species, within population approaches. But it has been demonstrated that a lot of the invasive species are exotic species that can adjust, like the urban environment works as a filter to select species that are easily adjustable to conditions. So I don't know about here in North America, but we have several species that have become invaders, especially in terms of birds, such as parakeets, and they probably have higher cognitive abilities and flexibility towards urban conditions. So this is not something I'm working on, but it's definitely a result that is out there in the literature. Thank you. Well, I think that there's no more, oh, maybe one last, because I'm not keeping track of time, but we have to finish before 15, so we've got 2 minutes.
Good morning. This should be a short question. Looking across your urban gradients within your different cities, have you quantified the amount of green space on that gradient and seeing if that correlates to the phenotypes that you see along the gradient? Because I imagine that would make them like closer to the forest, although not in the forest.
Yeah, thank you for that question, because I didn't detail how we measure urbanization. So initially in our project, we had kind of complex measure where we were studying, we were actually sitting there on sidewalks and counting pedestrians and counting cars. We also had the light pollution measured, and we had this vegetation index. But when we started upscaling to meta-analysis across a lot of different cities, we couldn't just go to 13 different cities and count pedestrians. So we have been using a much simpler approach, which is the ISA index, so impervious surface area. And this pretty much tells you how much green space there is compared to impervious surfaces. So this is really the type of measure that we use now, because it's easily measurable using satellite images, so you can do that in any kind of new area that you want to study. Yeah.
Thank you.
Thank you very much. Thank you everyone for all the questions, and see you around.